Can you see my screen?
Yeah.
Yes, perfect. Good. Welcome. Today's agenda, we'll hear from Francois about microbiome-based protection for optimized quality and safety of cold smoked salmon. Veronique will... I'm sorry, there's an agent here. Please turn it off. If someone has an eye assistant, please turn it off. Veronique is going to talk about biosolutions in the food industry. Joanna is going to talk about microbiome-based strategies for meat analogs. Fadi is going to show us I can't pronounce this, but SORF ML platform. And Angeliki will present antimicrobial effects of organo-essential oils under active packaging for shelf-life extensions and safety of various products. And we'll have time for Q&As and some feedback. And first we have...
Yes, I will share my screen. Can you see my screen?
Yes, we can.
Perfect. So then for the next few minutes, I will introduce the Novonesis company, explain why we have decided to be onboarded in the MICROORC project. And I will have a focus on a specific work package 3, because we are coordinating these activities. But first, a few insights about Novonesis. Novonesis is a quite new company, 2 years old, but it's one of the world leader in biosolution for the food industry. And it is born from the merge of two biotech company, Danish biotech company, Novozymes, which is quite well known for the enzyme part, delivering enzymatic expertise to the food industry and other industry, and Christiansen, mainly known to deliver food cultures for many food areas, among them fish, dairy, meat and beverages application. So Novonesis, as I've said, is interested in delivering biosolution, and we have two major business units, one which is related to food and health biosolution, and one related to planetary health. And we are delivering enzyme, culture, protein, yeast, probiotic, and HMO. But today we will mainly focus on the culture application. So when adding bacteria, probiotics, yeast, enzyme, or protein to food, you can have a high impact on the processing, but also on the product. When I claim you can have an impact on the processing, indeed, you can obviously increase the yield of your production. And we have some evidence that you can produce more yogurt out of milk, or more salami out of the same quantity of raw material. There are also some available data showing that you can lower the energy or the waste usage by adding the right biosolution. And you can obviously also work with raw material which have different qualities. You can also highly impact on the product quality. And here you can influence the texture of your food product. You can influence also the microbial quality, allowing to have a longer shelf life or more safer product. You can have an impact on the taste by developing more umami flavor, reducing sourness as an example. You can improve the appearance of your product. You can also, for some specific application, improve the nutritional value or show health benefit thanks to probiotics, and also develop product with a simpler label. And that fits 100% with the target of MICROORC project, and that's why we have decided to be part of the consortia working on this MICROORC project. In MICROORC project, we are mainly involved in work package 3, dealing with the application of biosolution to improve food quality of safety of 3 different food applications: smoked salmon, as Françoise has just shared, but also chicken, fresh chicken and marinated chicken, and plant-based meat alternative. And the work package is divided in 7 tasks. The first one dealing with an evaluation or an overview of the main available biosolution, which have already been tested in the 3 mentioned food application. Then we have 3 work package focusing mainly on providing or developing data to assess how biosolution can help improving shelf life of and safety of fresh chicken, cold smoked salmon, and plant-based meat alternative. And here we have a real holistic approach, meaning that we are assessing how we can improve the safety and the microbial quality, but we are also having a look on the taste impact of those biosolutions. We are also assessing or developing a strategy to evaluate how easy it is for food business operators to apply those biosolutions in real life in their factories, as an example. In the task 3.5, we have a strong focus on developing new prototype for plant-based meat alternative. We are working on burgers, and we are working on with pea and fava bean and soy. And our aim is to develop burgers which have less ingredients in their label, but which lead also to a better sensory experience. And then task 3.6 is dealing with gut health and how biosolution impact on the gut health. And the last task is to work with our partners, Lusavies, to develop product produced at factory scales. So the most involved companies and academics in this work package are Nofima, Ifremer, Porto University, and Lusavies for producing samples from chicken, but also, as an example, Primor for plant-based meat alternative. When dealing with biosolution in MICROORC, we are mainly focusing on 2 applications: food cultures and fermentates. So I think it is good today to remember you what are food cultures and fermentates. So you won't find any legal definition of those 2 biosolutions at the European level, but EFCA, the European Food and Fermentation Culture Association, has proposed its own definition for food culture. And food culture are defined as safe live bacteria, yeast, or filamentous fungi used in food production. Obviously, for bioproduction, we are mainly using or exclusively using bacteria, mainly lactic acid bacteria. And food culture are defined as preparation consisting of a concentrate of one or multiple species. But as you see, when we say concentrates, it means that in food culture, you have more than 10 to the 8 CFU per gram or per milliliter of bacteria, which are alive and which are safe. And in food culture, you exclusively find microorganisms and unavoidable metabolites and media component carried over from the fermentation. So it is not enriched in any metabolites produced during the shelf life. If we move to the fermentates, that's the second biosolution which is considered in the MICROORC project. There is, again, no legal definition, but you can find a definition from the Food Fermentation Germany Association describing fermentates are preparation in which you find metabolic products of microorganisms produced during the fermentation of those microorganisms. And Mathieu and all have proposed their own definition of fermentate, which are defined as powder preparation derived from a fermented product and which can be obtained by the fermentation of microorganisms. And here you see that in this fermentate, you find microorganisms, but only mainly the supernactant and the fermented substrate of the fermentation. In VP3, we have already reached several milestones. So we have a survey of available food culture and fermentates that can be used to produce safer food products. What we have discovered during this survey is that we have, if you look into the literature, you obviously find many works dealing with how biosolution can be used to fight against pathogenic bacteria, mainly Listeria monocytogenes, but there is a significant gap when it has to do with how food culture are efficient to fight against spoilage and how can they be used to reduce food waste. So hopefully at the end of the MICROORC project and FOODGUARD, we will be able to deliver more insights about how biosolution can also help reducing food waste and fighting spoilage bacteria. And another relevant milestones we have already reached in MICROORC is the publication in Journal of Food Production of a paper dealing with the evaluation of the efficiency of biosolution. And here we have taken a holistic approach, meaning we have taken into account, obviously, the efficacy of those biosolutions to improve safety, overall microbial quality, but we are also approaching the comparison of biosolution from a regulatory point of view, from a sustainability point of view, also within the European Union. So with this small teaser, I hope I will motivate you to have a deeper look and to follow the next result which will come out of those European projects.
Thank you, Veronique.
Thank you.
Joanna, are you here?
Yes. Yes. Good morning. Yes. Just let me. Are you seeing my presentation?
Yes.
Yes. Okay. So thank you. So today, just let me minimize this. Okay. So today I will present some of the work that we have been developing at UCP in the scope of the work package 3 of MICROORC project named Microbiome-based protection for optimized food quality and safety. And the work that I bring today, it's not working. Okay, has the objective to study the role of fermentates and food cultures in sliced meat analogs in extending their shelf life and also controlling the pathogen Listeria monocytogenes. So for the shelf life evaluation, we work with 6 different prototypes produced.
Sorry, I don't think the slides are shifting, so we're still seeing the first slide, but we're not seeing the.
Maybe if we switch to
we have the right
mode.
view. Yeah.
And now? No?
No. Should I share my
You're
screen?
sharing the wrong screen. You're sharing PowerPoint, not the presentation.
Oh, sorry.
Joanna, I can share my screen.
Yes, I think it's better, please. Sorry.
Okay, do you see my screen?
Yes, thank you so much. Yes. Yeah, you can, okay, the objective and yes, here. Okay, so to evaluate the shelf life, so we work with 6 prototypes produced with the partner Primor. And those prototypes were produced with 2 buffered vinegars and 3 cultured dextroses. We also work with the control without any of those fermentates, but with the synthetical chemical added. So along 28 days and also at 45 and 60 days of storage, we characterize those products in terms of physical, chemical, and microbiological characteristics, and also perform the sensory evaluation. You can click, please. Okay, so regarding physical-chemical characterization, it is possible to observe that pH was around 6 for all the prototypes in all the replicates and also a long time, and the same for water activity with values around 0.95 a long time for all the prototypes also. Please. Regarding microbiological characterization in terms of total viable counts, it is possible to observe that up to 28 days of storage, buffered vinegar A allowed lower counts compared to the control, but also for buffered vinegar B, cultural dextrose C, and also E, the counts were below or near the microbiological limit established. After 28 days, prototypes with buffered vinegars and also controlled dextrose C keep a similar or better result in the case of buffered vinegar A, suggesting potential to extend shelf life. Please. Regarding lactic acid bacteria enumeration, it is possible to observe that buffered vinegar A was the prototype that showed lower counts, followed by buffered vinegar B and also controlled dextroses C and E. Please. Sensory evaluation, we performed a difference from control test, and we found that cultured dextrose C sample differed the most from the control, with statistically significant differences in appearance, overall difference, flavor, and texture, and it was also the sample with the lowest mediums for flavor, odor, and texture attributes. Please. So, as a brief conclusion, 3 fermentates improved microbial stability of sliced meat analogs compared with control, and 2 fermentates, both buffered vinegars used, contribute to an extended shelf life without negatively affecting product characteristics. Please. Regarding behavior of Listeria monocytogenes, we perform challenge tests with the same prototypes, and we use a cocktail of 7 Listeria monocytogene strains adapted to 8 degrees, and also 3 different commercial food cultures. Please. We use 10 grams of each prototype. We put it into a cereal bag. We sprayed with each food culture, please. We also use prototypes without sprayed cultures, but with Listeria monocytogenes, and all of those samples were used as controls. And finally, we sprayed each prototype, contaminated each one with the Listeria monocytogenes cocktail, and those were used to test the Listeria monocytogenes behavior. So regarding yes, please. Click. Yes, so each bag was sealed and stored at 8 degrees, and along 28 days of storage, we perform microbiological analysis to count Listeria monocytogenes and also each food culture. Please. Here, and we have a short time to present. I will just present the results for Listeria monocytogenes. And on the first graphic on the left, we can see the behavior of Listeria monocytogenes alone. This is a logarithmic reduction graphic. We can see that for both culture dextrose C and D, they inhibit the growth of Listeria, but also buffered vinegars A and B allowed counts less than those found for the control. When we had food culture to each prototype, it's clear that occur a synergic effect, and for food culture 2, the inhibition for Listeria monocytogenes were greater than for the other cultures, with counts or numbers lower than below the detection limit of the enumeration technique. So please click. So combining fermentates with protective food cultures enhance the inhibition of Listeria monocytogenes across contamination scenarios. And these combined microbiome-based strategies supports safer meat analogs. Please. And as a final remark, just our results demonstrate that microbiome-based solutions can effectively extend shelf life and enhance the safety of sliced meat analogs. And this reflects the broader mission of the MICROORC project, orchestrating food system microbiomes to reduce food waste and increase consumer protection. Please, just my final slide, just a big thank you for the team who helped with this work, and for you to listen. Thank you.
Thank you, Joanna. And then we have Fadi.
Yeah, sure. I'll share my screen. Hopefully you can see my screen now.
Yes.
So we're going to go back to FOODGUARD and follow up on what Francoise has been presenting. We are talking about a platform, SourceML platform, which is basically tackling two things. Now, monitoring in real life situation during the production, we are relying within FOODGUARD on rapid and non-invasive sensors. So the idea here is those rapid non-invasive sensors, they need to have a machine learning algorithm that will take the spectral data and then convert it into either a sensory score, like we've seen from Joanna, or into a bacterial count, like we've seen in the previous presentation. And it has two sides. The first side is an automated machine learning ranking platform, where you basically test different devices against a bacterial count, and you would like to see which machine learning algorithm perform best with which bacterial count, or for which bacterial count for which product, and another layer which is a blockchain layer for real-time tracing and monitoring. If we have time, I'm going to speak about the blockchain layer, but for now, we're only focusing on the SourceML platform. So this is basically how it looks like. And the idea here is you can upload your experimental results by choosing to create a new experiment. So to choose a new experiment, I click on create new experiment. I have already added, for the time constraint here, a new experiment I would like to add to the platform. So first I give it a name and short description, where the experiment has been done, and whether I would like this data to be public, so everyone can see it on the platform, to be private, only me, or I can share it with someone. So I can share it with George or with Francoise, but it will be hidden from the other people using the platform. And then I will move on into the step of adding my experimental data, and the platform would expect you to add some raw data in an Excel or CSV file. So for example, this is some data from multispectral imaging. You can upload your CSV file from here. This is basically the raw data coming from the device. And you can add as many devices or as many analytical platforms as possible. Those are the ones that the platform is compatible with, or you can choose to add a generic data. So the platform would expect or would accept you to add some generic data as well. And then I move to the next step, which is uploading what we call the metadata. And the metadata can be basically what you would like your model to predict. So it could be a bacterial count, could be a sensory score, could be authenticity class if you would like to use it for testing food adulteration. Once you're happy with that, you click end, and basically here I can choose the bacterial count. There is one file I have forgot to add. It's added here. I click end, and then the experiment is on the platform. And if I go to my experiment, so this is basically the data I just uploaded. It has 3 analytical platforms: multispectral imaging, HPLC, electronic nose data. You can perform some simple multivariate analysis on your data from here, like a simple PCA plot. You can display your sample names here, or you can assign a color. So I can color them by the total variable count, which is on the PCA media, for example. That's basically to test to see if you see any grouping or any clustering within your data. But the interesting bit is to perform a regression analysis and to basically train a model to predict the bacterial count. So you can either choose a new model or a pre-trained model that you store on the system. So here I'm going to choose 2 of the 3 analytical platforms. I have added 2 bacterial count and a couple of machine learning algorithms here. Then you can choose any sort of pre-treatment you would like to do for your data and the number of iteration, how many times you would like your model to run. You can leave the algorithm on the default values that we can see here. And once you click end, it's basically going to do a grid search, trying all the algorithm that you have specified or against all the bacterial count within your file, and it will tell you exactly which analytical platform give you the best performance. That's probably going to take 5 to 10 minutes. No, actually, it's quite quick. So as you can see here, it gives you the results from the grid search. So which analytical platform give you the best accuracy. So as you can see here, for example, Videometer seems to be performing quite well. It gives you 84% accuracy for predicting total variable count and also 92% for the CSV. You can either click and save your models, or you can download a comprehensive analytical report of this grid search that you have performed. That's basically how it's going to look like. So it gives you which platform performs the best with which algorithm on the system. And you get some more details about the actual analysis, the performance according to the 5 iterations that you have performed, and also some additional results for the PCA analysis and so on. So that's basically how it works. I can save those models then. If I click on save the selected rows, then those models are going to become available. And the idea here is those devices can then be implemented online or at line throughout the production and distribution chain. And the ultimate goal will be I can use this multispectral portable multispectral imaging device, take a reading of my product. That reading is going to go to the platform automatically from the device and will be assessed against one of the optimized and validated models that I save on the system. And then on the device, I can see the results or the predicted bacterial count just by taking an image of my product. I think I ran out of my 7 minutes, so I'm going to stop now, and thank you very much for listening.
Thank you, Fadi. Next up, we have our final presenter, Angelique. Are you here?
Yes, I'm here. Let me share my screen.
Thank you.
Okay, can you see?
Yes.
Great. Okay, so hello everyone. My name is Angeliki Lukaiki. I'm a PhD student at the Agricultural University of Athens. So first of all, thank you very much for the opportunity to present our work here today. It was work that was done under the FOODGUARD project. So today we'll briefly discuss the application of essential oils in active packaging for improving the shelf life and safety of food products. And more specifically, I will present its use in two different food systems, feta cheese and seaborne fish. So in recent years, there has been an increasing interest in natural preservation strategies in food systems, mainly driven by consumer demand for clean label products. And at the same time, highly perishable foods such as fish and dairy are particularly susceptible to microbial spoilage. So while conventional preservation methods are effective, there is a growing interest in different approaches, including natural antimicrobial compounds such as essential oils, as well as their incorporation into active packaging systems. So the overall goal is to reduce microbial spoilage, maintain sensory quality, and ultimately extend shelf life using natural alternatives. So what we did to evaluate the application of oregano essential oil, we tried different strategies depending on the food system. In the case of seaborne fillets, two approaches were applied. The first involved modified atmosphere packaging, and the oregano essential oil was incorporated inside without it being in touch with the sample at a concentration of around 1%. And the second approach involved aerobic storage, and the fillets were wrapped in sodium alginate edible films with the incorporation of the essential oil in the film matrix at a concentration of 0.5%. Similarly, for feta cheese, samples were again stored under aerobic conditions and wrapped in sodium alginate edible films, including essential oil treatment. In this case, we also inoculated with Listeria monocytogenes to assess the antimicrobial effect on a commonly found pathogen in dairy. So the concentrations were chosen based on preliminary experiments so as not to have a negative sensory effect. And in all cases, samples were evaluated through microbiological and sensory analysis. And to further evaluate our results, spectroscopic techniques, including FTIR spectroscopy and multispectral imaging, combined with machine learning algorithms, were used in order to explore rapid and non-destructive approaches for spoilage assessment. So moving on to our key results. In the case of seaborne fillets, different trends were observed depending on the packaging conditions. Under MAP, the addition of essential oil resulted in a reduction of total microbial growth of up to 2 log CFU per gram, which also led to a shelf life extension of 48 hours. Under aerobic conditions, with the use of edible films, the antimicrobial effect was more limited and depended on storage conditions. Here, shelf life was extended by up to 24 hours, with a more pronounced effect at higher storage temperatures. Sensory evaluation indicated improved scores in samples treated with essential oil compared to controls, such as improved aroma, which in the case of seaborne, it's really important. And additionally, spectroscopic analysis from both FTIR and multispectral imaging systems combined with the machine learning models showed promising performance scores for the spoilage estimation in both experiments. And the R-squared metric was up to 0.88 in some cases, indicating potential for rapid assessment. In the case of feta cheese, the essential oil treatment resulted in the suppression of yeast and mold growth, which is particularly important in aerobic storage of feta. There was also a reduction of pathogen populations of up to 3 log CFU per gram, indicating a strong antimicrobial effect. Now, based on the sensory evaluation, shelf life was extended by up to 7 days at higher storage temperatures, with treated samples showing overall improved sensory acceptance for attributes like aroma, taste, and color. Again, here the spectroscopic analysis showed promising performance with quite sufficient metrics for spoilage estimation. So what we concluded was that the effectiveness of oregano essential oil is strongly influenced by the food matrix and the associated microbiota. Again, the method of application through active packaging systems like edible films plays a key role in its overall performance. Also, the combination of machine learning models with spectroscopic techniques can be an effective and non-invasive tool for spoilage assessment. So specifically, oregano essential oil does show potential as a natural preservation strategy. However, its sensory impact is an important factor to be taken into consideration, as higher concentrations may affect the aroma, flavor, and even texture of the final product, highlighting the need for a balance between antimicrobial effectiveness and total quality. So therefore, further optimization is required in order to achieve consistent and reliable results across different food and packaging systems in the future. So that's all for me, and thank you very much.
Thank you very much.